Welcome back to Miss B Biology. This is a revision lesson covering all of topic 3, infection and response. Students, when you are finished with this revision lesson, you should be ready for paper 1, topic 3. The only thing I'm asking you to do is to subscribe to my channel to help grow this teaching and learning community. And as usual, grab your book and grab your pen and let us begin. Let us first talk about communicable diseases, also called infectious diseases or pathogenic diseases. These are diseases caused by pathogens and can be passed from one person to another. Students, this is a common exam question asking you for the definition of communicable disease, and it usually values 2 marks, so make sure you say everything that is on the screen right now. Pathogens are microorganisms that cause disease. For example, bacteria, viruses, protists, and fungi. When we are infected with any of these pathogens, we tend to feel ill. Why? That is an exam question. We feel ill because pathogens reproduce quickly, produce toxins, and damage our cells. There are 7 diseases that you need to know in great detail, and 3 of these are viral diseases: tubercle mosaic virus, also called TMV, measles, and HIV/AIDS. Let's talk about TMV first. TMV is a pathogen that affects plants such as tomato and tobacco. Mode of transmission, meaning how is it passed from one plant to another? It is passed when infected leaves rub against the leaves of healthy plants, or if contaminated tools are used, or if workers whose hands become contaminated with TMV go on now to touch healthy plants. Signs and symptoms of TMV. There is a pattern of discoloration on the leaves as the virus destroys the cells. TMV causes destruction of chloroplast, resulting in stunted growth of plants. Let us now look at how TMV affects the rate of photosynthesis and also plant growth. Remember, the light that plants absorb is absorbed by the chlorophyll that is found inside the chloroplast. And if TMV causes the destruction of chloroplast, it means that less chlorophyll will be present. And if less chlorophyll is present, it means that less light is then absorbed for photosynthesis, resulting in less glucose being made. So that is how TMV affects the rate of photosynthesis. TMV can also cause plants to have stunted growth, so they're not growing to their full potential. How? Remember, less glucose is made. It means that less protein will be made, because protein is made from amino acids, and amino acids are made from glucose and nitrates. Nitrates are absorbed by the plants from the soil by active transport. So that's a domino effect there. How do we control the spread of TMV? There is no cure or treatment, but farmers can grow plants that are TMV resistant. Also, practice good field hygiene. Ensure that your tools are sterilized. If you have dealt with infected plants, your hands and your clothing must be washed. Let us now talk about measles. Measles is caused by a virus. How is measles spread? This is a common exam question valuing 2 marks. Measles is spread by inhalation of virus when someone who is infected coughs or sneezes. Signs and symptoms of measles would be fever and red skin rash. How we control the spread of measles? It has to be isolation as there is no treatment. Let us now talk about the virus HIV. HIV may cause the disease that is called AIDS. How is it transmitted? It is transmitted through direct sexual contact and the exchange of body fluids such as blood. So if drug users share needles, it can be passed on like that. If on-screen blood is used for transfusion, then it could pass on like that. And if a mother is infected and end up breastfeeding her child, it could pass on like that. Signs/symptoms of HIV. It begins with a mild flu-like illness. HIV also attacks the white blood cells. What is the function of white blood cells? We know that white blood cells are responsible for killing pathogens. So it means that if someone is infected with HIV, they will have less white blood cells. It means that their immune system is now compromised. How can we control the spread of HIV? So one, using condoms. Students, please do not say use protection. You'll not get marks for saying that. Make sure you say using condoms. Next one, do not share needles. Screen blood used for transfusion. And lastly, HIV positive mothers must bottle feed their child instead of breastfeeding. Let us now look at the bacterial diseases. There are two that you need to know about, gonorrhea and salmonella food poisoning. Gonorrhea is a sexually transmitted disease caused by bacteria. How is it transmitted? By unprotected sexual contact with an infected person. Signs and symptoms of gonorrhea. Thick yellow or green discharge from the vagina or penis. Students, you need to know this. This is not TMI. Another symptom would be pain when passing urine. Please note that there are some people who are symptomless. Antibiotics are used to treat gonorrhea. Why are antibiotics used? This is an exam question. This is because it is a bacterial infection and antibiotics are used to kill bacteria. Prevention and control of gonorrhea, using a condom and reduce the number of sexual partners. Bacterial disease number 2 is salmonella food poisoning. Salmonella are bacteria that lives in the gut of many different animals. Mode of transmission, eating food infected with the bacteria and food prepared in unhygienic conditions. Signs/symptoms of salmonella food poisoning: fever, abdominal cramps, vomiting, diarrhea, and dehydration. How do we control the spread of salmonella food poisoning? Chickens are vaccinated against salmonella in the UK to control the spread of the disease. Other ways the disease can be controlled is by 1. Do not eat undercooked food, especially chicken, and 2. Do not prepare meals in unhygienic conditions. How to control the spread of salmonella food poisoning from one person to another. Wash hands before preparing food. Wash hands after using the toilet. Clean areas where a person has been ill. Let us now look at diseases that are caused by fungus and protist. So we're looking at malaria, which is caused by protist, and rose black spot, which is caused by fungus. Let us first talk about rose black spot. Rose black spot is caused by a fungus, and this fungus mainly affects rose plants. Signs that plants are infected would be purple or black spots developed on the leaves. The leaves often turn yellow and drop early, and you know this is going to affect the rate of photosynthesis and the growth of the plant. Mode of transmission of rose black spot. The fungus is spread by wind and water. How can we treat and control rose black spot? Using fungicide, removing and destroying the affected leaves, and lastly, breed roses that resist the fungus. Let us now talk about malaria. Malaria is caused by a type of pathogen called Protist. The Protist is carried by the female mosquitoes in their saliva. Students, please note that it is not the mosquito that is causing the disease, but it is the pathogen that it is carrying. Right? So the mosquitoes carry the pathogen, and so they are known as vectors. Mode of transmission of the protist pathogen. The protist pathogen cannot pass directly from one person to another. It means that if people are infected with malaria and they sit close to you or they cough or sneeze very close to you, you cannot get the disease like that. Here is how the disease is passed on. Mosquito bites an infected person. The pathogen passes into the mosquito. The mosquito then bites an uninfected person and passes the pathogen to them, That is how the pathogen is passed from one person to another. The malaria disease is very serious because it causes recurrent episodes of fever, which could be fatal. The protist pathogen affects the liver and damages red blood cells. This could be a one marker in your exam where you're asked to name the organ that the protist pathogen affects, and this would be the liver. You could also be asked for 2 marks to explain why people with malaria are usually tired, and this is where you need to speak about the red blood cells. Remember, the function of the red blood cells is to transport oxygen around the body for aerobic respiration, and aerobic respiration results in the release of energy, right? So if red blood cells are damaged, it means that aerobic respiration will be reduced, meaning less energy will be released, and so people will be tired. How can we control the spread of malaria? The best thing to do is to avoid getting bitten by the mosquito. So we could sleep under a mosquito net that is sprayed with insecticide or wearing long sleeve clothing. We can also use an insect repellent to avoid being bitten. Another way to control the spread is to remove stagnant water to prevent the vectors from breeding. If you're unable to remove the stagnant water, all you need to do is to add some insecticide to the water. Travelers may take anti-malaria drug so that it can kill the pathogen if it were to enter the blood. Next topic is non-specific defense system. So in this topic, we're basically looking at how to prevent pathogens from getting inside the body. We're going to be speaking about the skin, the stomach with the hydrochloric acid, and also your breathing system, focusing on the nose and the trachea. Every day we come in contact with millions of microorganisms. The openings of our body, for example, our mouth and our nose, and any break in the skin, give pathogens a way to enter the body. Not to worry, because the body has many defense mechanisms to prevent the pathogens from entering. Let us talk about the skin defenses. The skin produces sebum, which is an oily antimicrobial substance that kills pathogens. The skin acts as a barrier to prevent the entry of pathogen. Point number 2, students, is very, very important. All right, so what happens when the skin is broken from a wound? If the skin is broken, the platelets in your blood help to form a clot which dries to form a scab and cover the wound until the barrier is restored, right? This scab now prevents the entry of microorganisms and also stop you from bleeding to death. Defense of the respiratory system. The air you breathe in contains pathogens. Not to worry, because the mucus and hair in the nose traps the particles in the air that may contain pathogens and prevent them from moving down to your lungs. Let us continue with defense of the respiratory system. Say, for example, some of the pathogen got past the nose, did not get trapped in the mucus, and ended up in your windpipe. Remember, another name for the windpipe is the trachea. These pathogens are on their way down to the lungs, but your windpipe is adapted in such a way to prevent them from getting into the lungs. Let us look at the adaptations. The trachea is lined with a tissue. This tissue is made up of cells. One type of cell is called goblet cells, and these produce mucus, and the other cells are ciliated. Remember, cilia is a hair-like structure that is always moving in this sweeping motion, wafting anything that comes into its path. So if the pathogen enters the trachea, what is going to happen? One, the goblet cells of the trachea secrete mucus, which traps the pathogen from the air, and then the cilia would waft the mucus to the back of the throat where it is swallowed. Let us now look at defense of the digestive system. So you swallowed the mucus just now with pathogens. These pathogens will end up in your stomach. Say you also had some food and the food was contaminated with bacteria. The food with bacteria will end up in your stomach. Please remember, your stomach contains hydrochloric acid, and the hydrochloric acid will kill the microorganisms from the mucus that you have swallowed, or if you had food that was contaminated. What if the pathogens get past the skin, respiratory, and digestive defense? What would happen? Not to worry, there is another defense system that is called the immune system. The immune system is the second line of defense. The immune system is made up of white blood cells. The main function of the immune system is to 1. Kill pathogens, 2. Neutralize toxins released by pathogens, and 3. Create memory cells for faster response on second infection. White blood cells, let's talk about them. There are 2 types of white blood cells that you need to know about. 1, the phagocytes, and 2, the lymphocytes. Lymphocytes. Lymphocytes are white blood cells that produce antibodies. Antibodies are protein molecules that bind to the antigen of pathogens and prevent them from infecting our cells. Antibodies are specific. What do I mean by that? They have a specific shape that is only complementary to one type of antigen. So say for example, you had some antibodies for the COVID virus. The antibodies would bind to the antigen of the COVID virus because the shapes are complementary. That same antibody, though, will not bind to the antigen of measles virus because the shapes are not complementary. Do you remember when you learned about enzymes and how specific enzymes are? Because their active site has a shape that is complementary to the substrate. It is similar to this. All right, so remember, antibodies are specific. Another thing about lymphocytes is that they create memory cells. Memory cells allows for a faster response on the second infection. And this is why many people believe that you cannot get a disease twice, because the second time around your immune system will respond so fast that you don't even know that you were infected. What is the function of phagocytes? Phagocytes carry out a process that is called phagocytosis. This is where the white blood cell or the phagocyte will detect the pathogen and move towards the pathogen and engulf and digest this pathogen using enzymes. Please do not say the phagocyte will eat the pathogen. The word is either engulf or ingest. Some white blood cells also produce antitoxins that neutralize the toxins released by pathogens. Antitoxins prevent the toxins from damaging your cells. The next topic is vaccination. Every cell has antigens on its cell membrane. Antigens are proteins. Each cell has a specific antigen. For example, we have 3 cells right here. That is the antigen for that cell. That's the antigen for that one and for that one. So the antigens are specific. If you notice, the shape of the antigen for each cell is different. Our body cells also have a unique antigen. So if these are cells from an individual's body, these orange circles now represent the antigen. Our immune system knows our antigen, right? So those are orange circles representing our antigen. These are the white blood cells of our immune system, and they are happy because they're in contact with antigens that they know. If a foreign cell were to enter our body, for example, bacterial cells with a different antigen, the immune system will interpret this as a threat and respond. Here is how the immune system would respond to the foreign antigens. The white blood cells, to be specific, lymphocytes, they would produce the specific antibodies needed to fight the pathogen. Remember, antibodies bind to the antigen of the pathogen and help to get rid of them. Some of the lymphocytes remain in the body. We call these memory cells. They can remain in the body for many years. If you were to be exposed to the same pathogen, your immune system would respond faster. It responds by making the right antibodies. These are made quickly to get rid of the pathogen. You wouldn't even know that you are infected. And this is when we say you are immune to the disease. Scientists created vaccines from the knowledge they have garnered about the immune system and how the immune system responds to pathogens. Vaccination is the introduction of a small amount of inactive or dead pathogen into the body. The presence of the introduced pathogens will stimulate an immune response. So the immune system will respond in the same way as when you were naturally exposed to pathogens. The white blood cells, to be specific, the lymphocytes, they will produce the specific antibodies needed to fight the pathogen. Memory cells will remain in the body for a long time, and if you were to be exposed to the same pathogen, your immune system would respond faster. If you have been practicing exam questions on vaccination, you'll notice that this graph comes up quite often. What they can ask you to do is to state differences in antibody production after infection compared with after vaccination. This is usually a 3-marker, so ensure you have these 3 points written down. Point number 1, the increase in antibodies begins sooner after infection. Let us look at the graph. After infection, look at how quickly antibody increase begins. And after vaccination, the line is longer. So it took a longer time for antibodies to increase in the blood after vaccination. After vaccination, students, memory cells are created. And so that is why when a person becomes infected, they will produce the antibodies quickly. Next point, more antibodies are produced after infection than after vaccination. Let us look at the graph line for proof. After vaccination, look at how far the graph line moves up. That's the amount of antibodies that are produced compared to after infection, right? Look at how far the graph line moves up, right? So more antibodies are produced after infection than after vaccination. Last point is that the number of antibodies remain high for a longer period of time after infection, right? So let us look at it. After vaccination, you'll notice that the number of antibodies, they begin to decrease, right? So not much was left in the body after a while. And so let us look now at after infection. The amount of antibodies remain high for a longer period of time after infection. So those are the 3 points that you need to write down if you're ever asked this question. Let us now talk about herd immunity. Herd immunity is when a large proportion of the population is immune to a disease. People who are not immune are less likely to come in contact with someone carrying the pathogen. For example, this person is not immune. This person is carrying the pathogen. The person who is not immune is less likely to come in contact with this infected person because the majority of the population is immune because they were vaccinated or they had the disease before and got better. If the number of people vaccinated against a specific disease drops in a population, for example, we're only seeing 2 people right here vaccinated, what is going to happen? The rest of the population would be at a risk of a mass infection. As you can see, quite a few people right here are infected with the disease. People who are not immune are more likely to come across people who are infected and contagious, right? These people, they are not immune and they are in contact with people who are infected. And the reason for that is because less people are vaccinated, less people are immune to the disease. The next topic is painkillers and antibiotics. If someone has a bacterial infection, they may experience aches and pain in different regions of their body. Painkillers are used to treat the symptoms of diseases such as aches and pain. The painkillers do not affect the pathogen, for example, the bacteria. So while you are taking the painkiller and easing the pain and the aches in your body, the bacteria, they will continue to multiply. Examples of painkillers would be aspirin and paracetamol. Most over-the-counter drugs are used to treat the symptom of diseases without affecting the pathogens. Let us now talk about antibiotics. Antibiotics are treatments that are used to kill bacteria. In your exam, students, please do not say that antibiotics get rid of bacteria. Please make sure you use the word kill. They kill the bacteria whilst in the body without damaging the body cells. Antibiotics can be taken as a pill, a syrup, or an injection. An injection is usually given when the person is really ill and the antibiotics need to get to the bloodstream very quickly. Penicillin was the first antibiotic discovered in 1928 by Alexander Fleming. Antibiotics have had a large influence on the world's health and death rate as bacterial diseases that killed people in the past can now be cured. Most antibiotics are specific to a particular bacteria. For example, this antibiotic right here will kill this type of bacteria, but it might not kill this one. Let us now talk about antibiotics and viral pathogens. Antibiotics cannot kill viruses, right? So it doesn't even make sense for doctors to prescribe antibiotics for viral infections. Viruses reproduce inside body cells, and body cells would be damaged in order to get to the viruses. Lastly, let us talk about resistant bacteria. What do we mean by that? Antibiotics that could once be used to kill a particular type of bacteria no longer does. So for example, this antibiotic was once used to kill this type of bacteria, and after evolving or mutating, the same antibiotic can no longer kill this type of bacteria. Why? The bacteria is now resistant. Scientists need to create new antibiotics to avoid going back to a time when millions of people died from bacterial infections. The next topic is discovery and development of drugs. Drugs are traditionally extracted from plants or microorganisms. Examples of drugs from plant. The drug digitalis is extracted from the foxglove plant and is used to treat heart problems. The painkiller aspirin is extracted from the bark of the willow tree. Students, you need to know these. These can be 1-mark questions. Let us talk about drugs from microorganisms. In 1928, a biologist called Alexander Fleming was growing and studying bacteria. He went on vacation but did not cover the petri dishes he was growing the bacteria on. When he returned home, he noticed that mold was growing on the petri dishes. He also noticed that some of the bacteria were killed. He called the substance that killed the bacteria penicillin because it was produced by a mold, a type of fungus from the genus Penicillium. Penicillin was later used to cure bacterial infection. Penicillin is the first antibiotic medicine. In later years, other antibiotics followed penicillin. They have been very successful in fighting bacterial infection. Let us now look at drug development. Scientists and doctors have been working over time to develop new medicines that can be used to cure diseases. The new medicines that are created must go through a series of tests and trials. Why? To check for efficacy, toxicity, and the correct dosage. When we speak about efficacy, we are looking at how well the drug works. For toxicity, we're looking at the safety of the drug. And dosage, we're looking at how much of the drug should be taken. Let us now look at testing of the new drug. The new drug needs to go through a series of trial and testing stages. This first stage is called the preclinical stage, and this occurs in a lab. The testing is done on cells and tissues. What are we testing for? We're testing to see how toxic the drug is. Are the cells and tissues damaged from the chemicals in the drug? If the drug passes this stage, it moves to the second stage. Stage 2 is also called the preclinical stage, and this stage also occurs in a lab. The drug is tested on live animals, so a known amount of the drug is introduced into the animal's body to check if it is safe. So a rodent is used, which would be rats or mice, and then a larger non-rodent animal, for example, rabbits. These animals are used because they are classified as mammals, just like us, and so it is believed that however the drug reacts in the mammal's body, the rats and the rabbits, it would react similar in our body because we're classified in the same group. If the drug passes the preclinical stage, it will now move on to stage 3, which is called the clinical trial, where a low dose of the drug is given to healthy volunteers to check if the drug is safe in humans. We're checking to see if there are any side effects. Are people vomiting? Are people having diarrhea? Are they having pain after using the drug? If so, the drug will not pass this stage. But if there are none of those signs or symptoms being experienced, then the drug will move on to the next stage, which is a wider clinical trial. In this stage, low doses are given to a small number of patients. So these are people with the disease. So what they're checking for, they're checking to see if the drugs treat the disease. If the drugs seem to treat the disease and is safe, then a larger group of patients are given the drugs to find the optimum dosage. Let us now look at an important concept, double-blind trial. Some volunteers are given the new drug and others are given a placebo. A placebo is a substance that looks like the real drug but does not contain the active ingredient. So we usually call this a fake drug. In a double-blind trial, neither the doctor nor the patients know who has the real drug and who has the placebo until the end of the trial. Why? To prevent or eliminate bias. This is the final concept. Publishing of the results. The results from the trial are published in a journal, and other scientists check the results for false claims. This scrutinizing process is called a peer review. This can be a one marker in your exam, students. Please move on now, students, to my video with 100 multiple choice questions on paper 1. Thank you so much for watching and all the best in your exams. Bye bye.